words of advice that you'd like to give us would be gratefully received. You're very welcome to vote for number 3 on the speaker list. At the back, there's a little box, and do put it in the envelope, put it on the box, or leave it on the chair, or if you're very electronically adept, you can go onto our webpage and even do it online if you prefer to do that. But your comments on how this has been received over the course of the summer will be very, very helpful indeed. Thank you very much for doing that. So, this evening's final presentation of the series is a guest lecture by Dr. Kevin Strange, the Executive Director of the Mount Island Biological Laboratory. Kevin did his bachelor's degree at University of California, Davis, master's also at Davis, before going up to Canada to University of British Columbia in Vancouver. Then did his postdoctoral work at the Laboratory of Kidney and Electrolyte Metabolism as part of the National Institutes of Health. Kevin moved to Mount Island 2 years ago, I guess, to take up the position, moving from Vanderbilt to University Medical Center in Nashville, Tennessee. I decided I would check up a little bit about him, and one of the interesting things is you can't find very much, which always means 2 things in my mind. Either he is very, very smart, and by not putting too much out there for people to find out about, or indeed he's very modest. And I actually think it's the latter. There are some keywords, and this one I'd like to take There are some keywords. Let's take a note of how many of these we hear this evening. I don't think we'll hear many of them actually. So, ion channels, epithelial biology, osmoregulation, membrane transport physiology, functional genomics, model organisms, and C. elegans. I'm sure we're going to hear some of those. The last one, right? That's great. Now, we have a really interesting talk to conclude the series. The worm in us all, that's the C. elegans, maybe. What our primitive ancestors tell us about human disease and aging. And it's a real great pleasure to welcome Kevin.
Thanks a lot, Graham, and I have to say it's a little weird to be standing up here. At Graham's suggestion, we actually launched one of these science cafes on MDI, and it's a much more subdued sort of setting. So what I'm going to talk about tonight is actually what I talked about on MDI, and I've talked about it a lot in many different contexts. And that's what I'm going to tell you about. It really has a lot to do with the Mount Desert Island Biological Laboratory. But what I'm going to try to convince you of tonight is that by studying organisms that don't look anything like us, and you'll see some examples of that, we can learn an enormous amount about human health, human disease, and the process of aging. And that's really what this talk is all about. I'm not going to focus on C. elegans. I'll give you a little bit of examples of that. But the whole talk really is about how and why we can learn about ourselves by studying these primitive organisms. Now, the whole talk starts with this one, this phrase up here. This was the title of a very famous essay written in 1973. Nothing in biology makes sense except in the light of evolution. And a full disclaimer right at the beginning of this talk. Everything I'm going to tell you about tonight has to do with that. So if you do not accept the overwhelming amount of evidence in support of evolution, nothing that I have to say tonight is going to make any sense. And my suggestion is go back and get yourself another drink. Maybe get me one, because it'll either be a very short night or a very long night. So this is how the talk starts. This essay was written by Theodosius Dobzhansky. Dobzhansky was a very, very famous, well-known geneticist and evolutionary biologist in the last half of the last century. Importantly, Dobzhansky was also a very religious man who considered himself, he was a member of the Russian Orthodox Church, and he considered himself a communicant of that church. So he had religious beliefs, but he was also one of the most important proponents of the theory of evolution. And what Dobzhansky said, many things he said in this essay, is he said that without that light, meaning evolution, evolutionary theory, biology, becomes a pile of sundry facts. Some of them interesting or curious, but making no meaningful picture as a whole. And this really is absolutely the key to understand what I'm going to be talking about tonight. You really have to accept evolution. I'm going to take you through some of the aspects of it. But this is really, I think, one of the guiding focuses of the way research is done at the Mount Desert Island Biological Laboratory. We always think about evolution when we're studying the biological processes, because thinking about evolution, thinking about the evolutionary history of those processes, always gives you much deeper insight. If you didn't have that, you would just have, as Dubzensky said, just a pile of curious sundry facts. So this is very, very simple what I'm talking about. The whole idea of evolution, of course, was launched, or certainly formalized and driven primarily by Charles Darwin. And one of Darwin's sort of central ideas was that all life on Earth is related through a single common ancestor. And that's very important. And in 1859, Darwin published The Origin of the Species. And this was the whole idea of natural selection, the idea that everything arose from a common ancestor. And throughout the course of evolutionary history, you gave rise to all the diverse species that we now see. So again, his idea was that at some point in time there was a common ancestor, and then through this process of evolution and natural selection, you eventually gave rise to all the life forms on Earth. Now, does anybody have any idea what this is right here? You probably can't see it very well, but that picture down in the corner? Any thoughts? I'm sorry? No, this is actually in a marine environment right here. Any thoughts? Oh, come on. Man, what do you think it is?
Could that be done in Australia?
Yeah, that's probably where that is from, yes. Stromatolites. So these are stromatolites. This is basically, these are actually living, or parts of them are living right now. But stromatolites are basically cyanobacteria. And fossils of these stromatolites have been found that are 3.4 billion years old. And what a stromatolite is basically a mat of cyanobacteria, and that they're all living together in this mat. They eventually die, that mat becomes mineralized, more bacteria grow on top of them, etc., and they form these sort of rock-like structures. So again, a biologist or paleontologists have found these stromatolites that are 3.4 billion years old. The Earth is thought to be about 4.5 billion years old. And it's been suggested that probably this common ancestor down here, this very primitive thing that we could call life, probably arose on the planet somewhere about 3.8 billion years ago. And this idea, the fact that there's a common ancestor for all organisms, was tested using bioinformatics approaches, statistics, mathematical modeling, etc. And this was published last year in 2010 in May in the journal Nature, which is considered to be one of the preeminent scientific journals. And what this guy, Douglas Theobald, concluded was that there was less than a 1 in 10,000 to the 860 chance that life did not arise from a universal common ancestor. Does anybody have any concept of how big that number is? That is 10 followed by 2,860 zeros. Okay, so it's a remarkable number. And to sort of put that into context for you, it's been estimated that in our observable universe, there's 10 to the 21st stars. So 10 followed by 21 zeros. It would take all the stars in 136 of our universes to equal that number. We can't prove this happened, but does anybody take bets in here, ever bet on anything, gamble? Okay, you go to a horse race, and if a horse in that race had a less than 1 in 10,286 chance of winning, I don't think anybody's going to bet on that horse. If you are, you're crazy. So we can't basically prove this. We can't go back 3.8 billion years and watch this happen. But all the evidence points to a very serious likelihood that this is indeed what all life came from. Some sort of single-celled, very primitive organism quite a while ago, billions of years ago. So this is how the whole talk really starts tonight. And the whole point of this is that all life arose from a common ancestor. Then we share all of our DNA with these organisms, even the simplest organisms we can find on the planet. DNA, of course, is what genes are made out of. Genes are basically blueprints for making proteins, and proteins are what make biology. Proteins get together, they carry out specific tasks, they build cells, they carry out the metabolism inside the cells, they allow cells to reproduce, organisms to reproduce, etc. So DNA gives rise to genes, they give rise to proteins, that's biology. So if we all arose from a common ancestor, we share our DNA with even the simplest of these organisms. And these are some of the organisms that scientists use very widely. Many of them we study at the biolab. This is a hydra up here and a very simple planarian or flatworm. These organisms are actually being used as very powerful models to understand tissue regeneration and wound healing, because they actually are remarkable at doing that. This little planarian flatworm right here, you can actually cut up into 300 different pieces, and each piece will grow a new worm. And scientists are now studying this to understand the genes, because we have many of the same genes that this flatworm has, to understand how that process of tissue regeneration and wound healing occurs. This is a very powerful organism here. It's a zebrafish, or as Graham says, a zebrafish, right? Yeah. A zebrafish. Very common a fish. If you have kids that have an aquarium, they probably have this organism in their fish tanks. This is widely used throughout biology as a model system to study all sorts of things. Development, regeneration, again, very powerful organism. This is a squid. This organism, much of the fundamental understanding that really formed the basis of understanding how our nervous system, how our brains work, really was developed with this organism. And Nobel Prizes were awarded for that sort of work. This organism down here, anybody idea what this is? Any guesses? How about you, Chuck?
Well, it looks like a fruiting body.
Yes. This is a slime mold. It's not even an animal. Would anybody in the audience be surprised if I tell you that we've learned things about schizophrenia and cancer by studying this slime mold? Would that surprise anybody? Yes? Well, the reason we've learned about cancer and schizophrenia from this organism, not even an animal, it doesn't have a brain, obviously, so how can it get schizophrenia? It has the same genes that we now know give rise or are thought to be associated with schizophrenia and parts of our nervous system. It has genes that we now know are important in the development of cancer. So again, nobody wants to believe it doesn't look anything like you and I. And maybe you do know some people who look like this, or act like it anyway. But because we have the same genes that this organism has, we can actually begin to understand how those genes function by studying these kind of very simple organisms versus a more complex system. Now this is my particular favorite organism. This is what we actually work on in the laboratory. This is the roundworm, Sianorhabditis elegans, or C. elegans. It's a nematode. And if I can actually start this. There we go. This organism is about 1 millimeter long, which is, I think, 30 to 40 times the size of the width of a typical human hair. It was established as a model system for basic biological research back in the early 1960s, and since that time there have been 8 or 7 Nobel Prizes awarded for work done on this organism, because it has shed very important insight into a variety of very fundamental biological processes. And I'll give you just a couple of examples here. This is Andy Fire. He's at Stanford University, and Craig Mello, who's at the University of Massachusetts. And in 1998, they discovered something in C. elegans called RNA interference. And I won't go into the biology unless people are interested in that later. But I will tell you that 8 years later, they were awarded the Nobel Prize for that discovery. And that is extraordinarily quick for a Nobel Prize. Usually it's twice to 3 times that period of time from the time a discovery is made until the time you're awarded a Nobel Prize for it. Now, does anybody have any idea why they get the Nobel Prize so quickly for that kind of work? Any ideas? Importance. The reason you get the Nobel Prize is because of the things you've discovered has wide-ranging impact. In this case, they won the Nobel Prize in Physiology or Medicine, which means it had wide-ranging impact on human health. And this whole process that they discovered, RNA interference, has given rise to 2 important things. First of all, it's given rise to a whole new biotech industry, because it's been recognized that this process of RNA interference may represent a very important, powerful new way to treat a variety of diseases, from cancer to you name it. There's a whole variety: heart disease, cancer, developmental defects, etc. The other reason that this is an important discovery is that the discovery of RNA interference actually led to the discovery of something called microRNA. And again, I won't go into that biology for you this evening. But microRNA ended up opening up a whole new way of understanding how our genes are regulated, how they're turned on and off. Completely unknown before. In fact, it's probably the most important discovery in molecular biology and biology in the last decade, is the discovery of these microRNAs. Now you might ask yourself, well, okay, they discovered it in this little weird worm. Could they have discovered it in a mouse or a rabbit or a rat, something that's a little bit more closely related to us? Yes, it could have eventually been discovered in there. But because of the simplicity of this organism, the economy of using it in the laboratory, the speed with which you can do experiments, it was discovered literally decades earlier than you could have ever discovered it in a mouse model or something more complex. So this is, again, one of the key powerful aspects of these sorts of organisms, is you can make discoveries much more rapidly, much more economically, and those can drive further studies in more complex organisms. Now, this is another great example of what was one of the key discoveries made in C. elegans. And I could go on for days up here going on about discoveries made in this and these other organisms. Anybody have any idea what this picture represents?
Aging.
Aging, right. Used to be thought 15 years ago, 20 years ago at most, that aging was a little bit like what happens to your car over time. It just kind of wears out. You can try to fix it here and there, but eventually you go buy yourself a new car. But a very enterprising scientist, a woman by the name of Cynthia Kenyon, about, again, 15, 16, 17 years ago, really began to question that. And she said, well, maybe there's a genetic basis. Maybe our genes and our DNA somehow control aging. And she thought that this little worm up here, C. elegans, might be a very good model to study aging and try to get at this question, is do genes control the aging process? And the nice thing about C. elegans is from the time you have a fertilized egg, and it's 3 days until you reach a sexually mature adult, and then after that, you got about 2.5 weeks of lifespan. So these guys live at most 2.5 to 3 weeks. And so if you want to study aging, doesn't it make a lot of sense to do something in an animal that only lives 3 weeks? You know, a mouse lives 3 to 4 years. So you're going to do an aging experiment in a mouse, you got to wait at least 4 years to start getting some data. This guy, you only have to wait about 3 weeks to start seeing the results of your experiment. So what Kenyon did is she started doing genetic experiments and starting to look for animals, mutants, that had longevity, were long-lived. And she ended up discovering a series of genes. One of those genes was called the FOXO gene. And the FOXO gene she found in C. elegans was regulated, or its activity was regulated by insulin signaling. C. elegans has the same insulin signaling pathway that you and I have. So we all know about insulin, we all know about diabetes and obesity, everybody's heard about that. Worms have an insulin signaling pathway. And that insulin signaling pathway regulates the activity of this FOXO gene. The FOXO gene, in turn, regulates the activity of a whole variety of other genes inside of cells. Those studies, when you have a certain kind of mutation in the FOXO gene, or you have mutations in the insulin signaling pathway, they give rise to worms that live 2 to 3 times longer than normal. Now that would be like you living to be 160, 220 years old. So it actually doubled and tripled the worm's lifespan if you had a certain kind of mutation in that FOXO gene. Now, scientists got very interested in that. They said, well, maybe this is just something weird with this worm, who cares? Well, sure enough, it turns out that mice and humans have the FOXO gene. So studies were subsequently done in the mouse, and they showed the same thing, that if you had certain kinds of mutations in the FOXO gene, or you altered insulin signaling in a certain way, you gave rise to long life. And importantly, you weren't just sort of looking like this and living longer. You weren't sort of decrepit. You were actually living longer like this. So you had healthy longevity. Now that got very interesting. When people found it in mouse, they said, well, does this occur in humans? And subsequently now, there have been studies carried out in people who live to be 100 or more. And if you have a certain kind of variant of that FOXO gene, a certain kind of mutation in the FOXO gene, and you have one copy of that gene, you got it from your mother or father, you have a 2-fold greater probability of living to be 100 or more. If you hit the jackpot and you got one copy from your father and one copy from your mother, and so you have 2 copies of that mutant gene, you have a 3-fold likelihood of living to be 100 or more. and healthy life. And I actually know several people on Mount Desert Island who are well into their hundreds, one in particular who's a benefactor of our laboratory. Remarkable. 104 years old, I can be having the same kind of conversation I'm having with you with her. Remarkably healthy, remarkably vibrant individual. So could you have ever discovered the FOXO gene and its role in aging by studying mice or rats or rabbits or chimpanzees or something? Yes, certainly you could have. But that would have taken decades and decades and decades longer and hundreds of millions of dollars or more to get to that conclusion. In C. elegans, it was literally discovered with a couple of months' worth of experimentation and probably a few $1,000s worth of reagents. So the whole point of this talk is that, again, we all share our DNA with the most primitive organisms. If we share our DNA, that means we share our biology. And so what we do as scientists is really start with these simple organisms first, use them to begin to uncover what the genes are doing, how they function, what the biology means, and then as we begin to understand those processes, we take them into more and more complex organisms. The beauty of these systems is they allow you to move quickly, they allow you to move much more economically than you could in certain other organisms, certain other models. Now, the sort of concluding part of this part of the talk is that what we're really talking about is all life rose from a common ancestor. Is what happens through evolutionary time is that evolution basically takes something that works, something that lives, survives, reproduces, and it adds more complexity to it, just through the process of random mutations and natural selection. So if you go back to that very first ancestor, whatever it was, 3-point-some-odd billion years ago, that was a pretty simple organism, pretty primitive. But it had beginnings of the basic biology that all of us share. And what happens is through evolutionary time, all that's been done is you add more and more complexity to that system. And to sort of illustrate this for you, this is an example of an evolution of fasteners. And this is actually a real example. This is fasteners that human beings have developed over the last 5,000 years. And one of these sequences up here is the actual correct sequence of evolution. I want to see if people can actually come up with what the correct sequence is. And what happens again is you start with a very simple working model or working plan here, one of these particular fasteners up here. And then through the course of evolution, in this case humans adding more complexity to the fastener, you become more and more complex, you're able to do more and more different sorts of things. So if you think about that idea for a minute, maybe we can just take a quick vote in the room. Who in the room up here thinks that this is the correct sequence? You're going from the most primitive to the most complex, the most recent sort of fastener. Who wants to vote on this one? No? Nobody? How about this one? Okay, a few hands. What about this one? A lot more hands. This is the correct one. Okay, so think about it for a minute. Just think about it for a minute. So this is a rivet. This was actually one of the first fasteners that human beings developed. It was about 5,000 years ago during the Brass Age. And you can see it's quite a simple sort of fastener. It just really consists of nothing more than a shaft and a head. And it was made out of a soft metal, brass. You couldn't tell that from the diagram, but a very simple sort of fastener. And then the next sort of fastener that sort of fell in that sequence was the nail. And what you can see here is, again, you have the same shaft, you have a head, but now you've added a point to it. What you've also added to this is you have to start making it out of a different metal. It's very hard to pound brass because it's very malleable and it starts to bend and fold and everything. So this was more complex, it's made out of harder metal. Then you go to the screw. Again, you have a shaft, you have a head, but you've now added a slot to the head, so you can turn it with a particular type of tool, screwdriver. And you've added threads to that allow you to drive it into things. So again, you've added complexity. And then finally, the most recent fastener up here is the bolt. Again, you have a shaft, you have a particular head that has a particular shape. It's got several smooth surfaces that allow you to use a wrench to tighten it. And again, you have a different type of thread on it. So the whole point of this is that what evolution does is it starts with a simple working plan. In this case, it was a rivet, or you could think of that universal common ancestor 3-point-something billion years ago. And then through the process of evolution, more and more complexity is added to this system. And then we can go to the last example here. Just imagine yourself that you're an alien that's just arrived here on the planet. And the planet you're from, you're obviously pretty smart because you managed to get here to this planet. But the planet that you're from doesn't have automobiles. And you come here to this planet, and let's hope you're friendly, but you see all these things, automobiles zooming around out there. And you want to go understand what these automobiles do. And you've got two choices. You go to some auto museum and you see this thing right here. I don't forget what this thing even is. But this is something called a Bugatti, which I have no idea what that is either. But they're obviously both automobiles. And you want to start to understand how these automobiles work. You have no idea, no clue. You don't know anything about it. Which one would you start to study first? This one or the other one? First one. Because it's the simplest. All of this, all the basic characteristics of this are derived from that simple one there. It obviously has wheels, it has a drive shaft, it has a steering mechanism, it has some sort of engine that powers it. And what's happened is through evolutionary time, in this case human beings adding more and more complexity, it's got much, much more complex. So what you start with is a very simple system first. You begin to tear it apart, tease it apart to try to understand the basic mechanics of it. And we're doing the same thing in biology. That's why we can study C. elegans or a zebrafish or a planarian or a hydra. We're all descended from a common ancestor. We share our genes, we share our DNA, which means we share our biology. It's just a lot simpler and easier to get at in a worm or a fish or a fruit fly than it is going to be into a mouse or a chimpanzee or even a human. So that's why we start with the simple, because evolution adds complexity. And we can start with these organisms because of their cost, their economy, their speed, and understand the basic biology. And then just like humans have added complexity to cars or to fasteners, as we start to build up our knowledge of the basic biology, we can begin to understand that complexity that's occurred through evolutionary time. So that's the basic take-home message there. And what's the time?
You've got another 15 minutes.
Okay. So I'm going to change gears just a little bit, and I'm going to talk about something else here. And the reason I want to talk about it is interesting. I gave a talk a few weeks ago to something called the Pot and Kettle Club on Mount Desert Island. And I won't go into all the weird sort of sorority-like things you have to do when you go into this thing. But I will tell you, it was a bunch of very wealthy, largely retired businessmen. And I sort of had to tell them something about the lab and what we do. And I had to give them a little bit of that biology I just gave you. But then I decided I'd tell them a little bit about why we do science and why science and education in science are so important. And the response I got to that was quite remarkable. So I decided that from here on out, when I give these kind of talks, I'm going to get up on my soapbox and I'm going to talk a lot about this because it's absolutely fundamentally important in where this country is right now and where it's headed if it doesn't start making some corrections. You know, we all know we're in very difficult economic times. We all know that the economy is in bad shape. We're in debt up to our eyeballs. I'm sure most people here are disgusted by the way our government sort of dealt with the recent debt ceiling crisis. I don't care what political spectrum you're on, nobody can agree with the fact that that was handled well. So we all know times are tough. And a lot of the conventional thinking when times get tough is you cut the low-hanging fruit. You get rid of things where there aren't big lobbying groups, where there's not a lot of money behind that. And those low-hanging fruits include research and education. So that's really the conventional thinking. Tough economic times, let's just go after what's easy to get rid of. And research and education are that. And this is why this is really dumb and very short-sighted thinking. It's widely accepted that in the last century, over half the growth in the U.S. economy can be traced to investments in science and technology. Much of that investment by the government. And what's remarkable is if you look at the number of people in this country who have degrees in science and engineering, currently it's less than 7% of the population. 100 years ago, it was far fewer. So it's a relatively small fraction of the country that's driving an enormous amount of the economic growth and wealth that occurs in this country. I'll just give you a couple of examples. One of my favorites is something that Eisenhower did in 1958. He formed what was called the Advanced Research Projects Agency, or ARPA. That eventually got renamed the Defense Advanced Research Projects Agency, or DARPA, in 1972. And what ARPA did in 1966 is it formed the very first internet, computer network. And it did that between a variety of research institutions because it wanted to save money so that all these people wouldn't have to duplicate resources and they could share computer resources. And this ARPA net, as it was called, ultimately became what we now know as the internet. And that internet currently generates what is estimated to be $130 billion in goods and services annually. Now, I don't know what the investment in ARPANET was back in 1966. I'm going to take a guess that it's probably a few tens of millions, maybe a couple $100 million. That's probably an overestimate. I'm just going to guess that. But that's not a bad investment to give rise to a $130 billion industry. A couple $100 million, $130 billion industry, not to mention all the enormous social change, good and bad, that goes with the internet. Another great example, it's how I make my living in part, is the National Institutes of Health. 15 of the 21 most important drugs that were introduced between 1965 and 1992 were developed using knowledge and techniques that came from NIH-funded research. Those drugs were involved in treating cancer, AIDS, hypertension, depression, on and on and on, anemia, herpes, etc. These launched a multi-billion dollar pharmaceutical industry. Hundreds of billions of dollars have been made off of the development of these drugs that were originally launched by NIH-funded research. Not only to mention the incredible impact that's had on human health and welfare. So, I think you can agree that the investments in science and technology had a huge payoff. But where do we stand in this country in terms of science education? And this is where it gets really troubling to me personally. This is a statistic, and you can get these, all sorts of statistics everywhere. This is a sort of a typical science score. And usually the U.S. is ranking somewhere in the bottom of the middle of these scores. This particular score, the U.S. was ranking 23rd, behind China, Finland, Hong Kong, etc., Singapore. So we're way behind. We're poor, and we're roughly in the lower middle part of the pack. This is one of the ones I found most troubling. This is the expenditures on education based on a percent of the GDP of the country. The U.S. currently ranks 54th in the world. I must say I didn't even know that some of these countries exist when I first saw this. Anybody know where Kiribati is? Any? No, I have no clue either. Kiribati is number one. We rank behind Kiribati, Lesotho, Yemen, Vanuatu. I think that was on Survivor, I think, one time. That's where I knew about Vanuatu. But the U.S. is 54th in the world. So we're pretty poor. And yet we're in an economy now where investments in science and science education are critical. Now, after I gave this talk to these wealthy businessmen, I must say the response was overwhelming. I got bombarded with questions. And there was a real sense in the room that these guys were really unhappy with this. That they were watching this country go from prominence in the world to becoming second-rate very quickly. And they asked me a lot of questions, why I thought this was, how we could remedy it, etc. Two things really struck me. One guy came up to me after this talk. He was a former executive at Pfizer. And he told me a remarkable story. He said that when he was at Pfizer, he used to work in the Maryland Pfizer, I don't know what city in Maryland it's at. And he said at some point during his tenure at Pfizer, Pfizer laid off about 6,000 people in Maryland. And he said simultaneously he saw the Pfizer group in Vienna, Austria increase by about 6,000 people. And he went crazy about that. He went up to the higher executives and said, what's going on here? And he says, the cost of living, the cost of doing business in Maryland is a fraction of what it is in Vienna, Austria. This makes no business sense. And the guy said to him, well, yes, it does. He said, in Vienna, Austria, we can hire kids out of high school who have the science and math and technology training we need to work in Pfizer. But in Maryland, we have to go out and find people with PhDs and master's degrees to do the same job. We have a hard time finding them, and we have to pay them a lot more money. It makes better business sense for us to go to Vienna, Austria, because the people are better educated there. They have more science and math education. Now, I went home that night, I was kind of amazed by that story, and I happened to turn on the news. I hadn't watched the news in about 3 or 4 weeks. It's the kind of jobs that Graham and I have. And there was a big segment on the nightly news about jobs and the economy. And the whole thing focused on high-tech jobs. 52% of the high-tech jobs in this country go unfilled at any time. They can't find qualified people to fill those jobs because they're not trained. They don't have the science, they don't have the math, they don't have the technology background and education they need to fill those jobs. We have over 9% unemployment now, and yet there are jobs out there begging to be filled. But we don't educate. We're not educating properly. We've cut education, we've cut science, we've cut the very things that help drive the country, while other countries are exceeding us. It was remarkable. Siemens, which is a high-tech manufacturing company, has, any given time, over 3,400 jobs open. And what they do, you have to get that job, you have to come in, you have to take an aptitude test. The aptitude test includes reading, basic science, and mechanical skills. Any guess on how many people, what percentage of the people who take that test fail it? How much? 90%. 90% of the people in this country who take a very simple aptitude test to work at Siemens fail. Because our high schools, our colleges aren't educating people in the proper way, and we're cutting science and education while we're trying to do this. So the last thing I'm going to leave you with, and I stole this, I have to admit I stole this. I don't mind plagiarizing as long as I actually acknowledge it. I was out at an event a couple weeks ago. It was with the National Park Service and the Scientifique Education and Research Center, which is there on Mount Desert Island, or just across the bay. And Susan Collins was there, our senator. Barbara Mikulski, the senator from Maryland, was there. Gary Mackless was there. He's the first ever science advisor to the National Park Service. And the whole discussion about that facility out there centered on science and education. And one of the things that I thought was remarkable that Gary Mackless said is that science and education are not luxuries. Science education, science research are not luxuries. They're absolute social responsibilities. Again, the jobs in this country can't be filled, the high-tech jobs, because we're not training people correctly. We have over 9% unemployment. Other countries are rapidly expanding science and education. China invests, I believe, something like 20% of its GDP annually in research and science. While we're cutting. So if we think about this country and think about what this country really means and what this country has been about, as Barbara Mackless said, one of what's part of our DNA, our social DNA, is discovery. She talked about how Thomas Jefferson sent Lewis and Clark out, and she said, you know, Jefferson didn't say to them, you know, go out and form the transcontinental transect, bird survey or whatever. He basically told Lewis and Clark, go out and discover. John Kennedy in 1961 didn't say, well, maybe we should try to build some sort of space-like craft thing that can go do something. He basically said, we're going to fly a man to the moon and bring him back by the end of the decade. And I can remember in 1969 sitting in front of my TV set watching the moonlight. But we've sort of gotten away from that in this country, while other countries seem to be surpassing us very rapidly. We've got a huge deficit problem, we've got a huge problem with unemployment, and we have all these high-tech jobs ready to be filled, but nobody there. So science and education are not just luxuries, and not just things that we should be doing in good times when we have lots of things to do. They're absolutely fundamental social responsibilities, and it's what this world is all about. And that's my soapbox. I honestly do this for a living, but I think it's for real. So I'll just stop there, and if anybody has any questions or want to throw something at me, I'll be happy to take them both. Questions, yes?
I remember somewhere between 10 and 20 years ago reading an article in Smithsonian about aging population and studies in Nova Scotia where there was an unusually high population of centenarians, and they concluded that it was genetic.
Yep.
And was the FOXO gene already discovered at that point, or?
The FOXO gene was probably about 15 or 16 years ago. in terms of understanding what it did and how it works in its role. And that really began with C. elegans. It was only, I want to say, probably about 6 or 7 years ago that the studies were replicated in mice. And then once they were discovered in mice, people got very interested in whether, we knew humans had the gene. It was very easy to find the gene in humans. But then the studies began in centurions and looking at their particular type of FOXO gene, the variant they had and whatnot. So that's been fairly more recent. That's probably been in the last 4 or 5 years. Some right here.
Is the research being done on these simple organisms that relates to Alzheimer's?
Oh yeah, absolutely. One of the things that you can do, I mean, C. elegans doesn't get Alzheimer's, obviously, it would be difficult to know. But you can actually engineer the worm. That's the other thing beautiful about some of these animals. It's very easy to genetically engineer them. In fact, we don't work on Alzheimer's in the lab, but one of the proteins we work on is called Huntington's protein, Huntington's disease, which is also a neurodegenerative disorder. And we have a worm strain that carries the mutant protein. And you can actually begin to understand the problems with the protein and what goes wrong with it. There are groups studying the protein associated with Alzheimer's and C. elegans and some of these other organisms. And the kinds of questions people are asking, the problem with Alzheimer's and Huntington's is that you have a mutant form of the protein. It's expressed in your brain cells, your neurons. And that as you age, this protein, which is nicely folded in a particular shape that functions properly, it starts to misfold, and then it starts to stick together with itself and other proteins and starts to form these big blobs and aggregates inside the cells. And those protein aggregates start to kill the cells. They kill the neurons. That's why you get the various defects. So what people are using organisms like C. elegans for, or Drosophila, is trying to understand what are the mechanisms that keep that protein in its proper configuration and keeping it from sticking to other proteins, and what happens when that starts to go wrong. Why, as you age, does that process start to break down, causing this problem? So if the animal doesn't have it, you can engineer it to make it do this.
First, I want to make a comment. You talked about the government being dysfunctional.
That's my opinion.
Yes. I'd like to point out that it accomplished exactly what its owners wanted, which is lower taxes on the very wealthy.
I would agree with that.
And in any case, I am curious about the FOXO gene.
Mm-hmm.
There are several of us in the room here who would like to live to be 160.
Mm-hmm.
And it occurs to me that evolution has gone on for billions of years without it being subject to controls by any of the participants.
Mm-hmm.
All of a sudden, we now have people like yourself and others that are jiggering around with these little worms, finding potential means for extending life. What happens when we get to the point where we can do that?
Yeah, that's a great question. It's called neo-evolution. I mean, medicine is evolution. I mean, once you started figuring out how to treat disease, wounds, etc., you were messing with normal biological processes in the course of evolution. So what happens? I personally don't believe we should be living to indefinitely, or I'm not even sure I'd agree to be 160 is a good idea. But what we'd like to be able to do is that you live like this, and then you start to decline, and all that takes a huge amount of input from the social systems, medical system. It's enormous cost. That's one of the big worries right now. I mean, our medical system is already overtaxed. We have an aging population, and obviously people want to take care of each other. They want to take care of disease and whatnot. So you keep putting more and more energy and money into sort of just barely living. You're on this declining stage. This is what you want to do. Live like this and then die. End of it. I think everybody would agree with that, right? So I think, scientists, the goal would be is that you live a healthy life, you minimize all the pathology, all the degeneration, and then eventually it is going to end. I mean, I don't think any of us, I don't want to live to be forever.
Well, how do you know how you feel when the time comes?
Sure. These are all the problems that it's like genetic engineering and everything else, or when you're, it's not too long, probably a couple of years, where it'll start to become economically feasible to routinely sequence everybody's genome when you go into a checkup. You want everybody to know what your genetic makeup is. I mean, there are benefits and downsides to everything, and that's a major social discussion that's going to have to occur at some point. But I personally think I'm happy to live as long as I can healthy. I don't want to live in a degenerated state for a long period of time.
I suppose that's hundreds of years.
That's, I'll be gone probably by then. It's somebody else's problem. But you're right. No, it's an absolute issue. I think we probably have plenty of people here on the planet already. So how do you deal with that? And that's a major question. But every time you discover something new, there's both a good side to it and a bad side to it. Somebody in the back.
Genetic engineering is a term that gets a lot of people wildly excited as being a horrible thing or whatever. And yet, for centuries, we've selectively bred and what's the difference between back 100 years ago when somebody selectively bred animals or selectively, we call it in biology when you join.
Where you're crossing, you know.
Cross-pollination.
Yeah, exactly.
What's the difference between that and what we call genetic engineering today?
Well, I think at least I sort of agree. First of all, I agree with you. It's actually a gradation. I mean, once you start crossing species or crossing different varieties of plants, for example, you are effectively engineering that plant to do something you want.
But maybe not without the controls that in engineering. There they try to experiment. Some failed, some didn't.
Right.
But
Well, I think the big problem that concerns a lot of people is you're actually putting foreign genes into an organism that might not have that gene. And that's the problem. And so if you cross two varieties of corn to get one that's more robust, they're still corn. If you now suddenly take a gene and put it into the corn that makes it resistant to some herbicide, they don't have any genes that make it resistant to herbicide, really. That's a very foreign gene you're putting in there. So people start to get very concerned about that. I think some concern is well placed. I think some of it's misplaced. But on the other hand, if you don't put the controls in place, people start doing all kinds of things. So I think it's the idea that you're really putting genes into that organism that there's really nothing similar to it in there to begin with. And you're really engineering it to start operating in a completely different way. And that's what a lot of people get upset about. And although I must say, I don't think they really understand it in that level of depth, though, to start with. I don't know who's next. Yeah, I think they're next.
I just remember hearing a couple lectures of sea urchins and the variability of their lifespan. I'm not sure, but it was like 4 months to 325 years.
Yeah.
And the crucial difference was the telomeres that get shortened. And so did the long-living ones have that FOXO gene? I never remember hearing about it.
Well, so two things. So you're absolutely right. There are, in fact, we have a scientist at our lab who just received an NIH grant to study that very thing. You got short-lived sea urchins, and like you said, that live months, and then other ones that live hundreds of years. And so scientists are actually trying to study them to understand what the differences are between the long-lived versus the short-lived. Obviously, you can't do longevity experiments on something that lives to be 300 years old. That's again why you see elements. Telomere length is an important component of aging. It's thought to be it's an active area of study. I honestly can't tell you that's what they found that or not. I don't seem to recall that, but you might be right. Do they have the FOXO gene? I'd be pretty stunned if they didn't. I'll bet you a beer that they did. I mean, again, C. elegans has it. C. elegans is more primitive than C. urchin. So. Yeah, I don't know who was next. Somebody in the back.
Do our primitive organisms like C. elegans or sea urchins, for instance, can they contribute to our knowledge of stem cell research?
Oh yeah, absolutely, absolutely. That little flatworm I showed you up there, that planarian, they have stem cells. And people are studying them to understand what it means to be a stem cell and what happens during the course of aging in those kinds of organisms. So absolutely. And zebrafish, very powerful system to study stem cell biology and other sorts of things. So yes, the answer to that is yes. C. elegans, a little bit more complicated, but people are studying sort of stem cell biology and that, more from the reproductive tissue.
Can you regenerate a planaria from a single cell, stem cell?
Not a single stem cell, am I right, Chuck? So in this, the planarian, it's not exactly 300, I think it's 270 different bits you can chop the worm up into. Each one of those bits has to have what they define as a single stem cell in it, at least. If you get a bit without a stem cell, it doesn't seem to regenerate. So it has to have the bits with the stem cell around it. But Chuck is right. You can take the nucleus, put it in an egg, an enucleated egg, and start a new organism.
Oh, so you can do it from a single nucleus as long as you have an egg to put
Yeah.
in.
Do tumors have this FOXO gene? You might have mentioned.
Sure, yeah. I mean, so all cells, you know, our cells have the same complement of DNA in the gene. I can't tell you whether the gene is active or inactive in the tumor. What the FOXO gene does is actually quite interesting. It regulates the activity of all kinds of other genes. And those other genes actually allow cells to cope with stress, environmental stress. So it could allow the cell to cope with temperature stress, oxidative stress. I'm sure lots of people have heard about everything you buy now is an antioxidant, right? You can buy ice cream that's got antioxidants in it. It doesn't matter whether you get 300 pounds after you eat it. It's got you know, so these other genes actually help the cell repair stress-induced damage, protect themselves from stress. And there's actually, let me make sure I understand it, there's actually an interesting observation that tumor cells may actually be more sensitive to oxidants, and rather than killing the tumor cells, they're more sensitive to oxidants, and actually there's some suggestion now that protecting yourself from oxidants may be a bad thing because the tumor cells may be more sensitive to it, and it may be killing them quicker. So when you start eating all kinds of antioxidants, now, again, that was one thing I read, I think, in Time magazine a while ago. So I don't go home and stop eating your antioxidants. But yeah, they certainly have those genes, but what it's doing, I couldn't answer that. Any other questions? There's one more back there.
I'm German, so you showed the statistics that certain kids at a certain age can have better science and math knowledge or reading knowledge in different countries. In the US it's not so good. So the same happens in Germany. And always when it happens in Germany, there's this huge outcry. You see it's a very national and regional topic. And every newspaper and every magazine is on the front page, it will be discussed. Not that the Germans have solutions, but at the same time when these tests happen, in the US, I do not see anything in newspapers and magazines or anything.
Right.
So
well,
why is this not on the front page?
Well, I think there is. I mean, certainly I hear about it. We are talking about this. Is why I just did it tonight. Okay? Yeah. I told you I was getting on my soapbox, and you know, and I've seen the response to it. This event I was at with Acadia National Park and the Scudic Education Research Center, which is again just across the bay from Mount Desert Island. The whole focus was on that. The politicians were talking about it. Of course, they're politicians. Why isn't it a major thing? Why isn't there more of a focus? I don't know, but I think what you're going to see is more and more of it because, as I said, this talk I gave, watching the jobs go away to countries that can have kids out of high school ready to take the jobs, seeing that we have 9% unemployment, but the high-tech jobs can't get filled because people don't have the kind of education. I hope we start to do this, but frankly, everyone in this room has a vote. And I think everybody in this room wants to be sure that their kids, their grandkids, the kids of their friends at least have as good of a lifestyle as you have right now. And if we don't do something, I think what we're going to see is once you start down that hill and pick up speed, it's pretty hard to stop and get back up. So it's got to become more prominent. And I'm a scientist. I obviously have self-interest here, but I hope I convinced you it's not just me keeping my job for a while.
Very much, Kevin. Now, I want to make 3 comments, really, a comment and 2 thank yous. First off, I thought Kevin's second part to his presentation, where he talked about the long-term economic projections of the value of science, are very valid. I just wanted to leave you with one fact I picked up over the last year. If we look at research and development funding that the state of Maine puts into its universities, its non-profit centers, and so on, averaged against the total value of the state product, we rank number 47 in the State of the Union. It's not a particularly great place to be as a state. If, however, you think about the value of the research non-profit institutions, including Mount Desert Island, Bigelow Lab, Jackson Lab, averaged against the state domestic product, in terms of bringing in federal dollars into the state for research and development, we are number 4. There's California, Florida, Massachusetts, and then Maine. The non-profit sector in R&D does contribute to this state, and it does that in a very valuable way. So that's my little bit of a soapbox. Great. Second thing is, I think Kevin's given us a fabulous talk to end the series in over the summer. Not only was the science provocative, but I think the messages behind it, the value of science, how elegant the science can be, but also some of the challenges in front of us. I think he did that very well. And before we thank him, I also want to thank all of you, some of you who've come every week for 10 weeks to come to this series. We have really, really appreciated your support for doing that. Thank you, everyone, and thank you to Kevin.